Hello, I'm pleased to speak today to you about network medicine and disease. So first, let's consider the question, what is disease? Its definition and understanding vary over time and place in history. Names of diseases have been of necessity abstractions. And as in other areas of semantics, what we mean by a term is best discerned by what we do with it. Historically, the disease has been viewed in different ways. The paleopathological record has its limitations. Ancient texts are a semantic minefield without true and clear meaning in modern terms. For example, what was the plague of Athens or the Antonine Plague? We stood on firmer scientific ground by the 19th century with the work of Semmelweis and Pasteur, Koch, Virchow, Lister, and Osler. And through the 20th century, the etiologies and effective therapies based on biochemical and molecular mechanisms were increasingly identified by reductionism over a broad range of diseases. Now, reductionism, as you know, is the basis for the scientific method. It was established in its earliest form by Democritus and the atomists in ancient Greece. But of course, Descartes developed it in a form we know well and use on a regular basis in scientific thought. We are in search of ever more refined associations that can ultimately define causation. The limitations of reductionism, though, have been appreciated increasingly in this era of omics and big data. Weighted genome-wide association studies, polygenic risk scores, and other statistical approaches do little to address the problem of complexity in biological systems, largely because they can't easily address the wide range of interactions among genes or gene products that exist within a life form. Now, of course, there's a long arc of reductionism in biomedicine that I touched on 2 slides ago. In this review of this notion of reductionism in medicine, Green and I used tuberculosis as an exemplar disease. Beginning in the 18th century, all one could say about the disease was that it caused a person to be sick, phthisis, with progressive disability and death. By the early 19th century, the organisms that caused it were identified, and by the late 19th century, the specific microbe was identified, with therapies based on understanding mechanisms of infectivity becoming more advanced through the late 20th century. But the problem, of course, is the disease is ever evolving, and we are in need, as we've become progressively more reductionist in identifying specific molecular causes of the disease, of reassembling those component parts to explore the subtle differences among different patients with the same disease. We have Osler and others to thank for the notion that we should think of one syndrome involving one organ causing each disease, which has helped us as a paradigmatic approach to disease, but clearly has its limitations. So this current century offers us the challenge of reassembling these reductionist parts that define disease. There is a changing scientific paradigm that we can use to approach this reassembly, and that paradigm involves the notion of networks of interacting molecular components. Most biological systems are complex systems that form such molecular networks. The quantitative approaches to analyzing these networks have been evolving, and you'll hear much more about those approaches throughout this course. Network medicine is then a highly interdisciplinary field that applies these approaches to pathobiology and treatment. And I show you the first text on this topic we published now several years ago, myself, Laszlo Barabasi, and Ed Silverman. And over time, there's been a greater uptake on the notion of network medicine as a true discipline, as illustrated, for example, by our own Channing Division of Network Medicine that Ed Silverman heads here in the Department of Medicine at Brigham and Women's Hospital and Harvard Medical School, and the more recently established network medicine entity at the University of Vienna, led by Jörg Menke. Now, network medicine can define disease without oversimplifying it, and can do so within all of its richly complex genomic and environmental context. And I'll try to give you some examples of those points. Beginning around 2010, published in 2011, Laszlo Barabasi and I developed this concept piece about what network medicine could be used to help sort out. And our hypothesis was that if one were to look at the comprehensive interaction network of all proteins within a cell or organism, within that network of protein-protein interactions, within that interactome, are subnetworks or modules that are associated closely with a specific disease. These disease modules then serve as the focus for our efforts at understanding the particular drivers of a disease or the drug targets for it. Now, one can derive these disease modules in a variety of ways, as we first showed in our 2015 paper in work led by Jörg Menke as a postdoc. But I'll give you a simple example here of how one derives a disease module for thrombotic cerebrovascular disease, as Rusheng Wang illustrated in this paper in 2018. First, one identifies the disease phenotype of interest, in this case, cerebrovascular disease. One then ascertains the disease network components, or so-called seed proteins, from the published literature or from unbiased omics screens of various sorts. One can then map these seed proteins to the interactome and identify the connector proteins that link the seed proteins together. As we show here in this proto-module, which is only a small part of a larger disease module that would be too difficult to visualize if I were to show it all. The seed proteins are depicted in blue and the connector proteins in gray. The resulting module defines the disease network or the disease module itself within the interactome network. So why do this? Why map these disease proteins or genes that code for them to an interaction network that is unbiased? Well, the main reason for doing so is that the interactome provides the missing links among disease-associated proteins within the disease module and pulls the whole set of proteins together in definable pathways that can govern some aspects of the phenotype. This module is analyzed statistically to demonstrate that it is unique and discrete and separate from an equivalent number of randomly selected proteins in the interaction network with similar connectivity. So one can do this for a whole host of diseases, as we and others have done, and use these resulting disease modules as a way to understand drivers of disease. For example, and I just give you this brief summary here in the interest of time with the citations if you're interested, I show you work from our group and others that highlight novel disease drivers identified through this kind of network analysis, ranging from the vitamin D receptor and IL-10 as drivers of preeclampsia, to JAK2 as a driver of certain subgroups of patients with hypertrophic cardiomyopathy, to NED9 as a driver of perivascular fibrosis and pulmonary hypertension, to PSMD3 a transcription factor that governs valvular calcification, to GAB1, a transcription factor that governs steroid-resistant asthma, and to NFAT-C4, a transcription factor that's important in type 2 diabetes mellitus. This kind of richness with respect to the complex interactions that govern a disease phenotype offers extraordinary opportunity for understanding disease mechanism and identifying specific targets for therapies, as I'll touch on in a little bit. Now, this is not to say that conventional approaches to disease like genetics are not helpful. In fact, just the opposite. Network medicine informs genetic determinants of disease. An example of that point is our work with Fei-Hsiang Chung in a paper first in 2021 and a more recent paper just published at the end of last year, in which we asked the question, are disease-causing mutations, in this case in germline diseases, are disease-causing mutations more or less likely to occur at protein-protein binding interface sites than exofacial sites? And in his analysis of disease-causing mutations from the HGMD, Fei-Hsiang was able to show that there was about a 4 to 6-fold increase in the likelihood that disease-causing mutations exist at protein-protein binding interfaces rather than elsewhere in the protein. This was also true for somatic mutations in most of the malignancies he studied in these same two papers, and I won't show you a slide on that point in the interest of time. So we can learn much more and have greater insight into genetic determinants of disease by understanding how such mutations can affect protein-protein interactions in these complex networks. In this way, network medicine provides a path to precision medicine, wherein we also want to distinguish among different subtypes of disease. And I'll give you as an example of this point work on hypertrophic cardiomyopathy in our group by Brad Marin and Andrei Semikin. In this particular study, we were interested in trying to understand what the differences were in expression of hypertrophic cardiomyopathy among individual patients, some of whom progressed to severe septal hypertrophy requiring septal reduction surgery, and others of whom were asymptomatic. Now, it's known that mutations in more than 11 sarcomeric proteins with greater than 1,400 variants are associated with the phenotype, but its penetrance is highly variable. And what most people don't recognize is that only about a third of patients, maybe up to 40% more recently, have sarcomeric mutations with the HCM phenotype. So it is a convergent phenotype about which we need to understand the genomic context to appreciate differences among patients. So here in this complex figure, you can see 18 patients who underwent septal reduction surgery. We obtained the tissue from those patients and did bulk mRNA transcript assessment, and then mapped the differentially expressed transcripts to their protein counterparts in the hypertrophic cardiomyopathy module to obtain individual HCM modules on these 18 patients. And you can see that even from this altitude, that the modules are all different patient to patient in terms of shape and density. But more importantly, when we began to explore pathways that appear to be most highly differentially regulated, we found that there were subgroups of patients with different primary drivers of the phenotype, one of which happened to be JAK2, a well-known target in certain malignancies, but not previously appreciated as having any role in HCM or in any other cardiovascular disease. So this is an example of understanding the nuances of genetically determined diseases by exploring the individual disease modules or reticulotypes of those patients. So one can think of network-based precision medicine as linking genotype to phenotype through individual disease modules or reticulotypes, which can in turn also be linked to other types of networks like endotype networks or clinical networks to give us more insight into disease outcome, prognosis, or therapeutic response. Now, network medicine also then facilitates drug target identification. And I'll remind you that our standard approach to drug target identification and drug screening really stems from the earliest work of Ehrlich in the early 20th century, who was a dye chemist at Bayer and really was the first person to perform a low throughput screen exploring drugs that might affect, in the first case, the spirochete that caused syphilis. Now, in his work as a dye chemist, he was struck by the tissue selectivity of certain dyes and reasoned that there might be specific interacting species, or what we came to understand as molecules or receptors for those dyes. So that notion of there being a specific receptor for a specific molecule, ultimately a drug, set the stage for what we currently use and what the industry currently uses for drug target identification. In the early days, of course, we had little knowledge of molecular mediators of drug effects, and we largely screened drugs on the basis of their effects on phenotype. So we presumed there was a ligand receptor interaction of some kind that, through some mysterious set of interactors, ultimately governed an output that we could measure that was felt to be important for the disease of interest. But of course, from a network-based drug target identification perspective, we now have the ability to identify many, if not most, of the interactors between a true ligand receptor interaction and its measurable output that is affected by a drug. We, in fact, can identify the drug target, which I highlight here in red. And that drug target identification gives us much more insight into how one might approach, in a more specific way, drug therapies. Network medicine also identifies environmental or exposome effects in health and disease. And how might that be? Well, at the simplest level, you can make the case that some epigenomic effects and post-translational modifications of proteins can, as a consequence, have direct effects on the function of those proteins or on the function of the genes that are transcribed into those proteins. So exploring those environmental factors and their consequences for the interactions of proteins in these interaction networks is another lens through which one can look at the underpinnings of network medicine effectively. And I'll give you an example of this point that is a little more directly demonstrable and analyzable, and that is from the perspective of foods that we ingest and what the consequences are of the molecules in the food supply. In a piece we published in 2020, Laszlo Barabasi, Julia Meniketti, and I conceived of this notion of the dark matter of nutrition and made the case that to date, epidemiologists have provided much useful information in compounds in food that affect risk of disease or may be beneficial in preventing disease. But the numbers of such compounds are rather limited. And at the time we put this slide together, there were about 188 that were nutrients that were known in the USDA database that could have direct consequences for health. But through Julia's work in compiling from a variety of sources all of the great pool and growing pools of compounds found within the food supply. We've been able to identify close to 140,000 such compounds. An example of how one might map the known and unknown dark matter compounds to a specific food is illustrated by garlic in this slide, where there are very richly 69 of the 188 known nutrients, but also over 6,800 of the 139,000 now mapped compounds to the food supply. So food is complex, and its small molecule species can also be used from an environmental and a drug development perspective as yet another application of network medicine to therapeutics. And an example of the working approach to this strategy is illustrated here, where we have two disease modules shown in red and yellow. We have approved drugs that are directed to an approved drug, which is directed to two specific targets. But we also have drugs that can be derived from food compounds or compounds within the food supply itself, which themselves have targets that can in turn affect potentially a disease module. These concepts were recently analyzed for a host of such food molecules and were summarized in part in the Annual Review of Nutrition paper that I cite in the last slide, as well as a New England Journal of Medicine paper that should be coming out shortly. So we can then think about network medicine and precision therapeutics in the following way, that in a particular disease, we have a host of patients who manifest the disease in different ways. There may be a common underlying endophenotype, a primary pathway that drives the disease, but it is influenced by the context within that pathway, within which that pathway is expressed and is operative, which in turn is further influenced by other genomic or genetic signatures, as well as by environmental factors that can influence gene expression or protein function. So the goal would be to develop a network-based analysis of individual patient reticulotypes or disease modules, and use our knowledge of both the static and dynamic behavior of those modules to derive therapies that are specific for subgroups of patients. Namely, those therapies are those which are guided by network-based biosignatures. So I'll end by answering this question: why network medicine? Why do we think that network medicine should be pursued as a definitive and very useful approach to disease and its therapy? Well, first of all, network medicine defines disease, at least theoretically, in all of its causal complexity. We're not simply looking at associations or at some disease drivers within a context of other associations, the interactions among which are unknown largely. Network medicine informs genetic determinants of disease. It also identifies potential drug targets. It can elucidate tissue-specific disease modules, which I didn't have time to talk about. And it can be used to define patient-specific networks and drug targets. And as such, it can serve as the basis, the fundamental basis for precision molecular medicine. So I'll end here and thank you for your attention and look forward to participating in the discussion later. Thank you.